Hello, good night, good morning, wherever you may are. So today I'm going to present some recent work I've done on LPMO, and I tried to match the title on what was written on your homepage. LPMOs will give you a little bit of an introduction. I'm aware that probably most of you know this enzyme already, also given the previous talk. But this is just to remind you that LPMOs are quite unlike the typical glycosyl hydrolases that we know. For instance, they have a very exposed active site. So this is really solvent exposed. In the active site, we find a copper atom coordinated by two histidine residues, so tyrosine in fungi, phenylalanine in most bacteria. And the enzyme actually attaches or likes to attach to polysaccharides, to crystalline surfaces, and act on those. So we need to have a reducing agent, so we need external reductants to make it active. This was probably also a reason why this enzyme remained undiscovered for so long. So LPMOs, as we heard, been discovered in 2010. So in addition, we need an oxygen-containing co-substrate that is either oxygen or hydrogen peroxide. But the mechanism, I will come to that in a minute, is still very much under debate. So why is this enzyme interesting for industry, for research? So first of all, LPMOs attack crystalline surfaces. So what they do, they decrystallize, for instance, cellulose or chitin. And shown here are AFM images of cellulose substrate. In the middle, we can see crystalline feature. And upon addition of LPMO, you can see that this is attacked and largely removed. So when, for instance, we mix LPMO with a trichoderma resi cellulase complex, you can see that the cellulose degradation proceeds much faster than without LPMO. Again, the reason is that LPMOs make the substrate much more accessible. I will not go into very much detail with the catalytic mechanism. I will just say that the LPMOs attack either the C1 or C4 atom in a glycosidic bond, thus producing either lactones or ketoaldoses. Interestingly, so what we have here is the beginning of catalysis. So initially, as I've told you, as we heard previously, we need external electrons to make this enzyme active. So we go from a copper 2 to a reduced copper 1 state, and only then the enzyme will interact with a co-substrate. So the original hypothesis was that they act with oxygen, hence the name monooxygenases, and then attack the substrate. Importantly here is that when we consider the interaction with oxygen, we need a second electron along this reaction pathway. And once the enzyme turns over, we end up in a copper 2 state. So here we again need to have an electron, oxygen binding another electron, and so forth. However, this is quite in contrast, the reaction with hydrogen peroxide does not require a second electron, but rather the reduced LPMO would react with hydrogen peroxide. And once catalysis occurs, the enzyme would leave the catalytic cycle in the reduced state. One important thing here is that the catalytic efficiencies with hydrogen peroxide are much, much higher than for oxygen, about 2 to 3 orders of magnitude higher, at least what has been published on these enzymes. One important thing that I would like to note is that LPMOs in the last couple of years have emerged in a number of organisms. So, for instance, originally LPMOs have been identified in bacteria. The highest diversity we probably find in fungi, at least from the LPMOs that have been characterized, where they degrade all the major plant polysaccharides. But however, as of recently, we also find it in less innocent bacteria that are pathogenic to humans. the recent publication. It's very exciting that the LPMO apparently mediates the survival of this bacterium in human blood. And I think this will be a very exciting research area in the future. We even find LPMOs in the viruses that are insecticidal, and here they seem to enhance the virulence. As of recently, we also find LPMOs in certain plants where they or seem to inhibit the development of insects. And ironically, we find LPMOs in insects, which use them to degrade chitin containing food. And here is like a little slideshow. We apparently have like a zoo of LPMO containing organisms. I especially like silverfish. So be nice when you see them. The two main research areas that I was busy with are, first of all, the activation of LPMOs. So the question, where do these enzymes get their electrons from? And second, I investigated the peroxygenase reaction of LPMOs, so the interaction with hydrogen peroxide. So out of convenience, I copied this slide from a recent review, and this concerns the activation of fungal LPMOs. And you can see here LPMO in the middle, surrounded by different ways to obtain electrons. It's very much like a molecular swinger party. It seems that LPMO can get electrons from a lot of different sources. So for instance, from lignin or lignin fragments. These are a pool of electrons. This is tightly connected to lignin degrading oxidoreductases that provide breakdown products, for instance. As we've heard, we find a lot of oxidoreductases in fungal secretomes. These feed on soluble sugars from cellulose, from hemicelluloses, producing oxidized sugars. And these can, for instance, pass electrons onto phenolic compounds derived from lignin, which act as mediators. So we have a relay mechanism, and these mediators can reduce LPMO. And some enzymes, we heard the case of the AA7, can directly interact with LPMOs to activate them. So one special interest of my research was the enzyme cellobiose dehydrogenase, which I would like to further introduce. And in this picture, we can already see quite a bit about the enzyme. It's very special because unlike the AA7 and most of the other GMC oxidoreductases that we find, it contains a second domain. We have a catalytic domain that's responsible for sugar oxidation, and we have a cytochrome domain containing a heme B that has no known catalytic activity, but it will just pick up electrons from the FAD and will then donate it somehow to the LPMO active site, leading to an interprotein electron transfer. So we find actually LPMOs in about 90% of wood-degrading fungi. So the majority of these organisms contain LPMOs, and among these, about 60% also contain CDH enzymes. So there seemed to be a quite high overlap. And I couldn't identify a single fungus that contains a CDH without an LPMO. And this adds apparently to a very nice extracellular electron transport chain. And since we have a lot of really nice cofactors, we can use spectroscopy to investigate these electron transfer reactions. And that's exactly what is shown here. When we put it into a stopped flow, we can monitor the reduction of the individual cofactors. So the first bump here would be the reduction of the FAD upon substrate oxidation. And only then we would observe the reduction of the heme B. So here we have a sequential electron transfer. So first the FAD gets reduced, then electrons are passed to the cytochrome domain. The rates, by the way, are highly dependent on the source of the enzyme and can vary quite a bit. But in any case, FAD here is always much faster than the cytochrome reduction. We can use the same kind of technique to monitor the electron transfer between the CDH and the LPMO. And again, when we mix reduced CDH with oxidized LPMO, we can see a very fast depletion of electrons from the cytochrome domain. So this amounts to a molecular rate constant of about 10 to the power of 6 per molar per second. So this is very efficient, and in any case, much, much faster than catalysis by LPMO. A couple of years ago, we were lucky to obtain crystal structures of the CDH. This was a very lengthy endeavor. It took a lot of different enzyme to really get crystal structures. But in the end, we got, was quite happy, 3 different crystal structures with 2 different enzymes. And of note, what we can see here, so the first 2 structures are from one and the same enzyme. And we can see that the cytochrome domain and the catalytic dehydrogenase domain containing the FAD cofactor are separated. And even more, we can see that the cytochrome domain here, for instance, in this crystal structure is facing towards the screen, and here it's facing away. So there is a lot of mobility in this system. And even more, we got a third structure where these 2 domains were really tightly docked onto each other. And in this particular structure, the cofactors were like only a few angstrom away from each other. That would allow, for instance, electron transfer between the FAD and the Heme. Now, it's really tempting to assume that, for instance, in such an open conformation shown here, the cytochrome domain is really accessible, could nicely interact with an LPMO molecule, whereas here in the closed structure, the cytochrome domain could accept an electron and then again move away to interact with LPMO. In a follow-up studies, we also tried to have a look at these electron transfer reactions in CDH. Just to give you a brief example, we did perturbation, we applied perturbation methods in which we reacted the enzyme under different conditions, that is increased pressure, increased viscosity, varying conductivity, and shown here, just as an example, is what happens to these electron transfer reactions when we apply a higher pressure. So if we go up in the pressure, we see that the FAD rate goes up. This is well, frequently observed for many oxidoreductases, but surprisingly, the cytochrome reduction goes down. It really seems that in this well, particular case, when we apply pressure, we restrict the mobility of the enzyme, and we reduce the electron transfer. So it seems that we have to allow a certain conformational freedom to really enable this electron transfer reaction to occur. So what we can say is that this well, electron transfer reaction is really linked to domain motions. It's one of the outcomes. Next, we also had a look at the possible interaction sites between LPMO and CDH, and I have to acknowledge the work of Christophe Laurent, who did extensive modeling, so in silico modeling, using a number of CDH enzymes and a number of LPMOs, tried to dock them onto each other. And what they saw is that there is no conserved well, residues where these enzymes could interact. There is no binding patch or something like that. It always seems to depend on a cofactor to cofactor interaction. The only conserved feature, however, was this tyrosine residue, which we found to hydrogen bond to the propionate arm of the Heme in simulation. So this hydrogen bond formed in most of the time. And when we removed this wild-type tyrosine, placed it by a phenylalanine or leucine, we saw that the electron transfer capabilities, they dropped dramatically. And this really seems to be well, a factor in this interaction. Also, over time, we monitored a number of different CDH and LPMO pairs and tested this electron transfer reaction. And well, we did that under varying conditions that contained the co-substrates, hydrogen peroxide and oxygen. We did it in the absence of hydrogen peroxide and in the absence of oxygen or hydrogen peroxide. And the important thing here is that these by molecular rate really doesn't change. So we end up whatever CDH LPMO pair, whatever condition we use, at the reaction rate of about 10 to the power of 6 per molar per second. So that tells me that, in fact, the electron transfer reaction is uncoupled from subsequent reaction steps. That is the first reaction step that occurs. Also, what we did here is that we monitored the electron transfer reaction in the presence of a substrate. And here we took advantage of one of our LPMOs that is able to convert a soluble polysaccharide. So siloglucan is soluble, is transparent, so we can use it with our spectroscopic methods. And that's exactly what we see here. So when we have a substrate present, the electron transfer reaction doesn't really seem to be inhibited. Only, I have to say, at higher concentration of siloglucan, we see a little bit of a drop, but also the viscosity of the medium changes quite a bit. But however, at the reasonable siloglucan concentration, the electron transfer reaction seems to be, well, very hardly altered. And one would assume that having the copper center, a site where electrons have to go in, and the substrate binding sites, that the substrate, if it binds, may inhibit this electron transfer, but this doesn't seem to be the case. In fact, what we observed and what has been noted for bacterial LPMOs as well is that, in fact, reduction of the active site copper also initiates or enhances the substrate binding. And this is a simple pull-down assay that we see here. So here is LPMO in solution, and that's what happens when we add a substrate. In this case, it was a cellulosic substrate. A fraction of the LPMO binds, and hence the intensity decreases. But if we add also a reducing agent, then apparently the binding becomes much stronger. So here we can assume that reduction induces binding to the substrate, and then catalysis can happen. That leads me to the second part of the talk, that is the peroxygenase reaction of LPMO. To investigate that, we chose a fairly simple method. We went for turbidimetry. To explain that a little bit, what we have done, we used cellulose. In this case, it was phosphoric acid swollen cellulose. You can keep that very nicely in solution. It's a rather homogeneous mixture if you mix it very well. So here we can see a quartz cuvette filled with such a cellulose solution. And here we have a stirrer bar. And here what we did is to add ascorbic acid as a reductant for LPMO. We added, of course, the LPMO, and then observed what happens with or without peroxide. And when we add peroxide, you can see that the optical density decreases. So what we can do, we can put this thing into a spectrophotometer and just have a look what happens when we add peroxide to this system. And what we see here is in fact the same experiment here. This is a solution containing cellulose, containing ascorbic acid, and the LPMO. And here I added stepwise fractions of hydrogen peroxide at different concentrations. So at each step I would add hydrogen peroxide, and you can see that the absorbance or optical density instantly drops. And if I double the amount of hydrogen peroxide, also the change roughly doubles from 20 to 40 to 80 micromolar. So we see a very fast, a quick reaction. So this change here is due to degradation of the substrate, and it really happened within the mixing time of this instrument, which is around about 8 to 9 seconds. An interesting aspect in this curve is the black curve here. And this is what happens when we just add ascorbic acid to the cellulose and the LPMO. And well, frequently this reaction is used to kind of determine the oxygen reaction of LPMO. So you have ascorbic acid, the LPMO, and your substrate, and you observe a reaction. However, if we carry out same reaction just in the presence of catalase, that would be here, the dashed lines. So here from bottom to top, we had the identical reaction just with different amounts of hydrogen ascorbic acid. I would see that catalase actually inhibits this reaction. So in fact, what we can conclude here is that the reaction with ascorbic acid is not an oxygen dependent reaction, but it's rather that ascorbic acid would produce hydrogen peroxide in a side reaction that initiates LPMO activity. Again, catalase in sufficient amount completely inhibits this reaction. Also, I think it's important to show this, again, as a kind of a confirmation. What we did here is to measure the rate, so the drop in optical density. So we took the initial slopes from these reactions and plotted it as optical density per minute. And we did 2 experiments. So here in blue, you can see a reaction where we added increasing amounts of LPMO, so from 1.5 to 6 micromolar, while keeping the amount of ascorbic acid constant. And you can see that if we double the LPMO concentration, the rate does not increase notably. However, when we maintain a constant LPMO concentration, but increase the ascorbate concentration, then we see a linear relation. So here, the more ascorbic acid we add, the higher the reaction. And in fact, this should not happen if we assume a reaction with molecular oxygen. By the way, the Km value of LPMO for ascorbate is in the low micromolar range. Here we are really saturated. Again, that shows that predominantly the reaction originates, the reaction with ascorbic acid originates from slight amounts of hydrogen peroxide being produced by the reductant. So having the knowing this peroxygenase reactivity of LPMO, we kind of went back to our beloved Cdh enzyme, and we thought, okay, so if peroxide is critical for the reaction, can we enhance the oxidase activity of cellular-biased dehydrogenase? Being a dehydrogenase, of course, you have a very low turnover with oxygen. So we went and did like a side saturation mutagenesis of critical amino acids around the flavin cofactor. And I will, well, cut a very long story very short. We identified a combination of mutants that was really sufficient to increase the oxygen turnover of this enzyme. And what you can see here, the wild type is an inefficient oxidase, whereas our variant enzyme has a 30-fold enhanced oxygen turnover. Importantly, the electron transfer capabilities to LPMO did not change. And so we set out to use this variant enzyme for LPMO reaction. And we chose a bacterial LPMO just because it's easier to monitor the reaction products. So what we can see here on the y-axis are reaction products by the LPMO, and on the x-axis, the concentration of the CDH. And in fact, 1 hour into the reaction, we can see that our variant enzyme really outperformed the wild type. So with much lower concentration of CDH, we produced much higher concentration of product. And after 24 hours, so your reaction is finished, still the very similar picture is observed. With minute amounts of our variant enzyme, we achieve a very high concentration of product. So if we compare, for instance, here, using 13 nanomolar of the CDH, gives us the same product concentration as 1 micromolar of the wild type. And we also tested this with LPMO that's active on cellulose. Here, reaction products are much more difficult to quantify. but just looking at the chromatograms, we can see that using the same low amount of enzymes here, the variant enzyme gives a much higher product formation than the wild type. So in fact, what we have created is an enzyme that is able to donate both the co-substrate and the electrons to the LPMO reaction. Lastly, and very briefly, we also had a look at the stoichiometry of the peroxygenase reaction, and just briefly recap the hypothesized reaction mechanism of LPMOs with hydrogen peroxide. We can see that LPMO is reduced, will bind to a substrate, will interact with an hydrogen peroxide molecule, will leave the reaction cycle in the reduced state, and is then right away ready to bind the next hydrogen peroxide molecule. So in fact, what we need is just a simple priming reduction, and then the enzyme is able to carry out multiple reaction cycles. And to put that to the test with our fungal system, we used here a cytochrome domain from Cdh as an electron donor. So we can nicely discriminate between the reduced state of the enzyme and the oxidative state. So we can quantitate the amount of electrons that we have in the system, which is really nice. So we next, and this is a very scary looking graph, we combined our reduced cytochrome domain with an LPMO and added hydrogen peroxide to the system. And what we observed was that if we add a substrate for the LPMO, then you need a lot more peroxide to bring it back to the reduced state. So without substrate, this is a purely stoichiometric reaction. You need the same amount of hydrogen peroxide to deplete this cytochrome domain from electrons. But if you initiate catalysis, then you need a lot more hydrogen peroxide. And this translates into this graph. It actually shows that for a single electron, or that with a single electron, our enzyme can turn over about 20 times. And lastly, we tried to confirm this also using spectroscopic methods. And for instance, Epr spectroscopy, we can see Epr spectrum of here an oxidized LPMO that we've seen in the previous talk. When we add a reducing agent in stoichiometric amounts, we end up with this spectrum here. So these spectral features largely disappear. And an important thing is that when we start with a reduced LPMO, when we add, for instance, here hydrogen peroxide to it, we will completely reoxidize the enzyme. So here, reduced LPMO plus hydrogen peroxide gives us oxidized enzyme. Importantly, this is not observed when we have a substrate there. So when we allow substrate turnover, we add again our soluble substrate for the LPMO, and then add the same quantity of hydrogen peroxide, we again see that we end up in the reduced state. And this I also did with UV spectroscopy, very painful because you need a high amount of LPMO to visualize the tiny LPMO peak. But however, same picture. If we have a substrate there, addition of hydrogen peroxide to the reduced enzyme doesn't lead to reoxidation. So in fact, to conclude here, we could show that reductive activation regulates the biological function of LPMOs. So despite having a number of different reducing agents available, it seems that LPMOs largely rely on partner redox enzymes that can be regulated, probably on a genetic level. We could show that fungal LPMO have a dominant peroxygenase activity. So I would say driving remaining questions are that a lot of electron donors for instance, bacterial LPMOs need to be identified. And it also has to be put to the question whether hydrogen peroxide is a universal substrate. If all LPMOs have access to hydrogen peroxide, this will also be a question for future research. with that, I would like to end and thank a lot of people. So most of this data was generated in the labs of Roland Ludwig and Nigel Scruton, where I spent exciting times. Also would like to thank Vincent Eising and his group, and Peter Mann, Peter Halleda in the Czech Republic for support, and yeah, sponsors and universities. Thank you very much.
Thank you, Daniel, for a really nice talk. Excellent science. And I'm glad you said that some of the experiments were painful because, I mean, yeah, they do require quite a bit of protein. We have some questions here, and I first goes to Leila Loldeggio. If you can unmute yourself, then you can ask your question, Leila.
I don't have a question.
Okay.
Okay.
I don't know.
Maybe you
Sorry.
do.
Well, maybe then I'd have to think very fast. I can ask one anyway.
Yeah, I mean, it's your show.
Well, I mean, since it's my show, but really, it's first of all very nice to see your work. But the other thing that I would like to say is do they need to be regulated?
No idea. So
because aren't so many of the LPMOs extracellular? I mean, enzymes that are extracellular, are they usually regulated? And if they are regulated, why?
Well, of course. So the reason, so when we look at fungal genomes, you find a multiplicity of LPMOs. So some organisms contain like 10, 20, 30, 40 LPMOs. And one reason is probably that these LPMOs, of course, have different substrate specificities. It would make sense for a fungus to kind of activate, produce an LPMO with a certain specificity if needed. So the case, for instance, Neurospora crassa has LPMOs that are active on cellulose, on soluble oligosaccharides, on starch. So of course, a starch LPMO only makes sense if you have starch in the environment. So yes, I strongly believe that they're regulated. Also in terms of CDH, we see substrate well, preferences.
Sorry, maybe I was unclear. I didn't mean regulated at the genetic level. Obviously, they are regulated at the genetic level in response to substrates, or at least I agree with you there. But it's just you suggested that the redox flux regulates them. Or maybe I misunderstood what you meant.
Okay, no. Well, that is, I think, well, up for discussion because I don't know. But because we have to look at that probably in a biological perspective. So at which stage of growth or in which environmental conditions, we don't know which reducing agents are available in nature. That's what I meant.
Okay, then I agree with you.
I'm glad to hear, Leila. First, there's a question from Bernard, then there is Maher. A question. So Bernard, if you unmute yourself, you can ask your questions.
Oh, okay. Why was CDH so easy to improve? Well, I wouldn't necessarily say it was easy. Well, why wasn't it improved by nature? Probably it was not necessary. I don't think that LPMOs or CDHs are meant to work under maximum possible conditions. They just have to be sufficient. I think the aim of a fungus might not be to degrade the substrate as fast as possible. That's why. But easy to improve, I disagree.
Okay.
Why was trichoderma boosted upon LPMO addition? Good question. Maybe by just adding more LPMO. Does trichoderma actually contain LPMOs naturally?
No.
No.
That's what I said.
Okay.
Yes,
well,
two.
Okay.
I'm sorry, but
two.
yeah,
But it seems
not
to be
many.
very poor. Somehow it's very poor in LPMO activity. Maybe it's because it has these exquisite GA7s. Maybe I don't know.
Yeah. Mahar, you have a question?
Yeah. So thank you very much, Daniel. Very nice talk, very inspiring. So my question to you is, have you done any work on, or do you have any theoretical answer to the specificity of the cytochrome domain in electron transfer? Because, I mean, Vincent has characterized also another GH or AA12 that has a cytochrome domain, and from the open conformation, this domain is wide open. And from the thermodynamic point of view, I would presume that it's able to carry out electron transfer to a variety of acceptors. So have you done any work to look at the specificity there?
I'm not sure if it's really, well, the specificity comes, so partly at least from a redox gradient. From my knowledge, what we've did, it doesn't matter which cytochrome LPMO pair you use, you end up with roughly similar electron transfer rates. So it seems to be redox gradient driven.
I see. My question is more in an in vivo context where you have a ton of other electron acceptors. How would you appreciate the specificity of how would the cytochrome would you characterize it as specific in transfer, in relaying the electron to the LPMO, or what it would hit anything it faces when there's a thermodynamically favorable electron gradient?
Yes, so I wouldn't say that it's highly specific. There are a number of electron scavengers around, and I would think that the LPMO would also transfer electrons there. Maybe one thing is that so both or many LPMOs and many CDHs, they can attach to cellulose. Now, chances are that these enzymes are close to each other during the degradation process. But how they maintain specificity, and if, I don't know, and well, at least I don't think they're only transferring electrons to LPMOs.
Thank you.
Lots of quinones around that could also be potentially reduced by the enzyme.
Okay, then we have Ivana. She wants to ask a question. So you can unmute yourself, Ivana. So you can unmute yourself now, Ivana, if you want to ask a question. Okay.
There's also a question in the Q&A, Morten.
Yes, I just saw that. Concerning the experiments with ascorbate, yes, that is.
This is from Steve Withers.
Referring probably to that graph, dependence of rate on LPMO concentration. Yes, so I think that the rate is pretty much limited by how much peroxide is available. So at the fixed ascorbic acid concentration, you would only produce so much hydrogen peroxide, and hence the reaction rate doesn't increase. That would be my take.
What kind of family do you look at here, Daniel?
That's AA9.
A9. So basically what you're saying is that for the first case on the blue line, I'm sorry, the black line, it's what they call the auto-oxidation of ascorbic acid.
Yeah. So if you throw ascorbate into buffer, chances are that you will produce hydrogen peroxide. And actually we have measured that also electrochemically. So there will be hydrogen peroxide being produced. And that serves as a co-substrate for the LPMO. Hence, when you increase the ascorbate, reaction rate increases. But when you maintain a certain ascorbate concentration, it doesn't. No matter how much LPMO you throw in, or at least it's very.
Yeah, I can do some advertisement here. You could look at a paper in Biotechnology for Biofuels 2021 by Stepnov et al., which this is really well investigated. So that was a question from Steve Withers, and now Vincent, you're next in line.
I was wondering, Daniel, nice to see you, by the way.
Hi.
I was just wondering, what is your opinion about the relevance of the oxidase activity of LPMOs in biomass degrading ecosystems? I mean, the ability of LPMOs to produce H2O2. Is that a relevant?
Of CDHs or LPMOs?
Is that sorry?
You mean CDHs or LPMOs?
No. I mean the LPMOs themselves. You know, if you have plenty of LPMOs and there's little substrate, they will produce H2O2, right? I mean that oxidase activity, is that relevant, you think?
Maybe, yes, but also I think that this reaction only happens if you do not have a substrate. And now I might sneak in another slide because what we have measured, and this is very confusing, so if you just mix a reduced LPMO with hydrogen peroxide, you will produce amino acid radicals. And that has been first observed by Jones et al. Very wonderful publication, I have to say. And they saw that when you react LPMOs with hydrogen peroxide without a substrate, you generate radicals, and these may take away oxidizing species from the active site. So they're kind of an emergency mechanism to prevent auto-oxidative damage. Whereas when you have, for instance, a substrate present, that's shown here, on the dotted lines, nothing happens. So I think that it's dangerous for LPMO to produce peroxide because that may also lead to auto-oxidative damage and indicates probably that there is not enough substrate around. I would say that at least in fungal systems, there is always, or probably a lot of peroxide around. For a lot of oxidases, you have peroxygenases that depend on peroxide. I don't think they need to produce peroxide by themselves.
I think the reaction is also much slower, but we can, yeah. Okay.